Okay, I'll start again. Hi, everyone, and welcome to today's edition of the 3D Regulatory Genome. It's my great pleasure to welcome somebody who I've never actually met in person, one of several speakers I've never actually met in person in the series, but whose work I've followed and admired for a long time. And it's a great pleasure to welcome and host Axel Visel today, who will speak about from nucleotide to megabase scale enhancers in developmental disease. Axel did his PhD in 2004 at the Max Planck Institute in Hanover in Germany, and then I believe moved right away to Berkeley from there. Is that correct, Axel? Where he to the Lawrence Berkeley National Laboratory, where he still is today as a senior staff scientist and deputy director of science at the Joint Genome Institute. And in addition, he holds an appointment as an adjunct professor at the School of Natural Sciences at the University of California, Merced. And Axel has made similar contributions to the enhancer field for at least 20 years, probably more like 25 by now, including he was one of the driving forces behind the Vista enhancer browser, which is one of probably the largest database of such kind, and that's just one of many other key contributions to the field. And yeah, with that, I'll hand over to Axel, who is it's 8 o'clock his time, so first talk of the day for him. Thanks a lot for joining us.
Okay, good morning. So let's see, let me just see if I can figure out my screen sharing. Okay, I assume you see my title slide now?
Yeah.
Okay,
and it's
perfect.
in presenter mode and everything, so good to go.
In the right mode, okay.
Yeah.
Got it. Okay, then I'll just go ahead. Yeah, so good morning, everybody, or good late afternoon if you're in the UK or elsewhere in Europe. I don't know if anyone is joining from Asia. In that case, good middle of the night, I guess. But so yeah, thank you, Stefan, for inviting me to give this seminar. And I will talk about my favorite topic, which is, of course, enhancers. So what I'll talk about is I'll start with a little bit of historical background about enhancers, why we should care about them at all, and go over some of the historical challenges that we've had in finding enhancers in the genome and characterizing them. I'll go over some solved problems, but then most of the time I'll spend on issues that continue to be challenges. And I go over some recently published and some unpublished work. So I will start the main part with some work that we published, I think 4 years ago, led by Evgeny Kwan, who developed new systems for developing or for assessing the in vivo activity of enhancers at scale, improved mouse methods. And then I'll talk about a study that we published just a few months ago, led by Michael Kosicki, who used some of these exact methods to look at enhancers in more detail, and in particular get closer to understanding the inner anatomy of enhancers, what parts of an enhancer are important for in vivo function. And then hopefully, if I can stay on schedule, I'll have time to talk about a third project that is not yet published, that doesn't talk about individual enhancers, but takes a step back and looks at these vast non-coding spaces in the human genome, so-called gene deserts. And I'll try to make a case that these are probably really important and that we should pay attention to them when we think about human genetics. So with that said, let's start with a little bit of general background. So I think we are, let's see. Okay, I think we are at a really exciting time in human genetics, in a sense that with the progress in sequencing technology, it is now very easy to sequence the genome of any human individual. Hundreds of thousands, or probably millions at this point, of individual human genomes are becoming available. So that part is really easy. But what continues to be a challenge is linking genotypes to phenotypes. What this means is that in the context of disease, that we understand how changes in an individual's DNA cause disease, or if they're not causative in isolation, how they change the susceptibility to a disease, or how changes in the genome influence general traits such as, say, height or the shape of your face. And I would argue that we've made pretty good progress in terms of understanding this for protein coding sequences. Still, it's not perfect, but at least we have pretty good leverage. But it continues to be a major challenge for the non-coding sequence space. And what we are interested in is, I mean, there are many different types of functions in the non-coding sequence space. One function that we are interested in are these distant acting enhancers, in particular enhancers that are active as the organism develops prenatally. And these enhancers are, I think, quite interesting. So just to give you a reminder what these are. So these are short sequences that you find scattered all across the vertebrate genome. So they're found in all vertebrates, including humans. And what is special about them is that they drive gene expression from a distance and with very high spatial precision. This is illustrated here in this cartoon. We have here a mouse embryo with a hypothetical gene expressed in the brain and in the limbs. And this cartoon enhancer has a limb enhancer. This cartoon gene has a limb enhancer and a brain enhancer. These enhancers can be upstream or downstream of the gene. They can even sit in introns. And what they do is they act on the promoter in a tissue-specific manner to activate transcription. So here we have the limb enhancer, and here we have the brain enhancer. And so in a limb cell, the limb enhancer would be active. In a brain cell, the brain enhancer would be active. At the molecular level, again, this is a somewhat simplified view, but what happens in principle is these enhancers contain binding sites for transcription factors. In the case of the limb enhancer, for transcription factors that are expressed in the limb. So in a limb cell, this would happen. Transcription factors bind, co-activators bind, a bunch of other things happen, such as histone modifications, but the ultimate outcome is that the enhancer interacts with the promoter and activates transcription. And in a brain cell, we would find the same thing, except it would be this enhancer over here interacting with brain-expressed transcription factors, co-activators, and then the same RNA is made. So this is how it works in principle. And this general phenomenon of enhancers has been known for 3 or 4 decades. So it's not a new phenomenon, but something that really became clear only over the last maybe 15 years, I would argue, is just how many of these enhancers are in the genome, the complexity of this entire framework. At this point, we know that we have at least 100,000 enhancers in the human genome, depending on how you define them. There may be hundreds of thousands. Keep in mind, we have about 20,000 genes. This means, on average, each gene has multiple enhancers associated with it. At the same time, we know that we have at this point mapped many of these enhancers, but not all of them. We still don't know where all of the enhancers in the human genome are. For the vast majority of them, we don't understand what they really do in vivo, what is their biological function. And this, in combination, creates a major problem in this new era where we can easily sequence entire human genomes, because what typically happens is that we find a lot of non-coding sequence variants. We can read out what happens in terms of the sequence, but it's really hard to interpret. So this is illustrated here in this cartoon, which is, I'd argue, like a pretty common problem in actual clinical genetics. So you have a patient with a disease that looks like it's, based on circumstances, like it's a genetic disease. And the first thing is you look for coding mutations, right? You can do this looking for gene panels, or you can do exome sequencing. And then in many cases, you find mutations in genes that cause this disease. But then in many other cases, you don't find this. All you find is a whole bunch of non-coding mutations. It can be de novo mutations or rare mutations. If you look at de novo mutations alone, every human has about 50 to 100 de novo mutations in their genome compared to their parents. But because 98% of the genome is non-coding, just statistically speaking, all but one or two of these mutations fall into the non-coding sequence space. And you commonly encounter situations like this, where you have point mutations or like small mutations that are near a gene that you know is implicated in the phenotype that you observe, but you can't quite make sense of this. And the reason why you can make sense of this is because you don't know if it changes anything. You can hypothesize that this is what we like to call an enhanceropathy, meaning a disease caused by a mutation of an enhancer. But to really figure this out, there are two important questions that you need to answer. The first one is you need to figure out if your mutation hits a regulatory sequence at all, meaning you need to know where the enhancers are. And then the second big question is, if it falls into an enhancer, does it change the function of this regulatory sequence, the enhancer? Does it change that at all? And so let me talk about these two questions separately. We'll first talk about the first one, because this is one, I think, where there has been great progress over the past 15 years, like figuring out where enhancers are in the genome. This used to be for a long time a hard problem because we don't understand the sequence code of enhancers like we understand the sequence code of protein coding genes, where we can look for consecutive stretches that contain triplets. But we made significant inroads towards this starting about 15 years ago with the development of ChIP-seq, which for the first time enabled the genome-wide mapping of enhancers that are active in a tissue or cell type of interest. So this was one of the early studies that we did on this, where we demonstrated that you can apply this ChIP-seq method directly to primary tissues from embryos. These could be mouse embryos, as shown here. These could be human embryos. This could be human adult tissues. The idea is you collect the primary tissues, you then cross-link proteins to DNA, you shear the DNA, and then the trick here is you use an antibody that targets enhancers only when they're active. In our early studies, we used antibodies for P300, which is one of these co-activators that sit on top of enhancers. You don't have to know the actual transcription factor. You can put on many different kinds of enhancers with these co-activators. Today, most people, including ourselves, use, instead of P300, use histone modifications that are associated with enhancers. But again, these histone modifications like histone 3K27 acetylation are present only when the enhancer is active. The bottom line is always the same. You pull down the DNA fragments that are essentially the enhancers that are active in the tissue of interest. You can then strip off the protein and then isolate just the DNA, sequence these, map them back to the genome, and this gives you these genome-wide coverage maps where each of these peaks is presumably an enhancer active in the tissue that you put into the assay. Now, that's great, except it comes with some caveats. The two major caveats are that none of the marks that has been looked at to date is perfect. All of these ChIP-seq marks generate both false positives and false negatives, which means some of the peaks will not validate as real enhancers in vivo, and they miss some enhancers. So what we like to do is we like to couple these genome-wide methods with in-depth validation using a true in vivo method, which is transgenic mouse assays. What we do for this purpose is we take any sequence that we think might be an enhancer, couple it to a minimal promoter, and then LAC-Z reporter. We generate transgenic mouse embryos by pronuclear injection, as shown here. So the construct integrates in a stable way into the genome, in this pronucleus. We can then put this back into a pseudopregnant mouse and then collect the embryos at whatever stage we're interested in. Most of what we do is around mid-gestation, as you see here, day 11.5 of the mouse pregnancy. And then we can stain these for LAC-Z activity. And then we see, for example, something like this. In this case, the forebrain turns blue and multiple independent transgenics. So we know now this is a true in vivo brain enhancer. So just to give you a broad sense of how these two different methods align, this is again from our first attempt of doing this. We collected 3 different tissues. We did ChIP-seq. again, this was in the early days of next-gen sequencing. So these numbers of reads that you get genome-wide are not that impressive by today's standards, but they were sufficient to get thousands of peaks genome-wide, most of which were specific to just one of these tissues. And we then compared this with results in transgenic mouse assays. So here just like a few examples to illustrate what this looks like. So we can, for example, take this region of the mouse genome that showed us like a nice P300 peak in the forebrain, but not in midbrain and limb. We clone it, we put it in a mouse, and it turns out this is a highly reproducible forebrain enhancer. Two more examples. Here's a midbrain peak that corresponds to midbrain enhancer, a limb peak corresponding to a limb enhancer. Some of them had activity in more than one tissue. Here is a forebrain and midbrain peak, and indeed we observed this kind of compound pattern. So these are just a few examples, but what we observed is when you go for the like really highly significant peaks using this approach, in the majority of cases, you get nice agreement between the ChIP-seq predictions and the in vivo assay, which means that this now provides us with a method that can be used to find enhancers at scale, in particular enhancers active in a tissue that we're interested in, for example, because it's involved in disease. So we applied this subsequently to various other tissues. So for example, to heart tissue. So here you see examples from studies where we used mouse and human heart tissue to perform ChIP-seq, and then we looked for enhancers that are not active, in this case in the embryonic heart, but in the heart at 4 weeks of age, so almost adult for a mouse. You see here two examples of whole mounts of the heart, and we can look at these in more detail. We can look at cross-sections of these hearts. So here are 8 different enhancers, and this really illustrates why it's important to do these transgenic assays, because this highlights another important caveat of these tissue-derived data sets, which is you can't quite tell in which cell type or subregion the enhancer is active. So this adds a lot on top of just having a ChIP-seq peak. Here's another example where we applied the same approach to forebrain tissues from the mouse embryo. So these are E11.5 mouse brains. We collected ChIP-seq data, and then we looked at the forebrain, which is already quite differentiated at this point of development. Here are just 14 of about 140 enhancers that we tested using this approach, and you can so this is just like one slice each. We did like full sections for these, but you can probably appreciate from this that these enhancers drive this expression with very high spatial precision. Here's a third example of a study. So these are craniofacial enhancers. And this is a study that was led in our lab by Katja Atanasio, who I think I saw on the participant list. So hi, Katja. So Katja looked at the craniofacial enhancers, again, using ChIP-seq from craniofacial tissues in the developing mouse embryo, and then mapping them using transgenics. And what you can probably appreciate even here from this low resolution whole mount image is that they drive, again, expression in very different parts of the developing phase. Now, these are just like 3 examples of publications. You don't have to look at these publications if you want to find the enhancers. We have a convenient database called the Vista Enhancer Browser that can be found here at this URL, enhancer.lbl.gov. And here we deposit all of the results from our published studies. And you can browse this for your favorite gene. You can browse it for your favorite tissue. And just last year, we released a major update for this Enhancer browser that provides improved support, for example, for time series of developmental time series of enhancer activity that allows you to compare directly reference and allelic versions of enhancers where we have tested those. And it also makes it easier to browse through various evolutionary alleles of enhancers that we have in our system. So if you're interested in this, I encourage you to go to enhancer.lbl.gov. And again, so this is relatively new. So if you have any suggestions for further improvement of the new UI, we are very interested in that. So with that, I want to switch gears from how can we find and define the the natural expression pattern of enhancers to the second problem that I talked about early on, which is understanding how sequence changes in an enhancer potentially affect its activity, which is, of course, what would potentially cause disease phenotypes resulting from enhancer mutations. And this has historically been very difficult. So in theory, enhancers are just like collections of transcription factor binding sites. And in theory, if you just find the binding sites and figure out which of them are mutated, you should be able to predict this. But it turns out that predicting the functional impact of mutations in enhancers is really hard. For a long time, I would argue we have been really bad at this. More recently, there has, I'd say in the last 2 to 3 years, with the introduction of a new generation of deep learning methods, there has been major progress in this area. We're becoming much better at this, but still, I would argue that at this point, in silico methods are not good enough that you would want to rely on them, for example, in a clinical setting to interpret a sequence change that you see, especially if the interpretation would have any actionable consequences in a clinical setting. I would argue that you will always want to have some experimental validation. Now, on the experimental side, there is at this point, a number of massively parallel reporter assays that can be used to look at enhancer mutations at scale. So they can look at hundreds or thousands of enhancer mutations in a single experiment. The problem with these is that they have high throughput, but because they usually need to be performed in cell culture, there is a limited number of cell types that you can even look at. And even when you do, they usually don't capture the full complexity of in vivo processes. For example, morphogenetic events that are the exact events that are disrupted in many structural birth defects. So for that, you usually need to look in vivo. And so that brings me to what we like to do in our lab, which is use in vivo methods, which give you the maximum amount of information in terms of experimental validation, but they have historically tended to be slow, expensive, and low throughput. I showed you earlier how we do these transgenic mouse experiments, but even with all optimization, you're still limited to maybe doing a few hundred of these. So that's about the throughput that we can do in our lab, but a few hundred of these per year. And so with that, I want to talk a little bit more, like go into some more detail about these transgenic assays and how this has been historically done and how we have recently improved on this. So the majority of transgenic experiments that were done until about 5 years ago, and still many of the older experiments that you find in the Vista Enhancer browser, relied on a classic transgenic assay that was introduced in the late 1980s, which is a reporter assay where you clone these enhancers with a minimal promoter and a Lexi reporter, and then inject them, and then random genomic integration happens. And so you can look at the embryos and get results. And so there are some advantages to this, which is it's a true in vivo method. So it captures developmental complexity. It gives you true spatial and temporal activity patterns, and it's relatively fast from injection to result. If you look at an E11.5 mouse embryo, you get there in about 2 weeks. But then you see here in this example also some of the disadvantages of this approach. So the first one you don't actually see, but it's hinted at here. So it's a random integration into the DNA, which means it's the effect of the efficiency is relatively limited. Of the embryos that you inject, only 10 to 15% on average turn transgenic. So you need to really inject a lot of eggs to get a good number of embryos. The second is shown here, and that's hinted at here. So when you do these random integration, then you get position effects, meaning that the construct integrates into a random position in the genome, and often it will pick up the activity of other regulatory sequences that may be present at the insertion site. These could be other enhancers that cause ectopic activity. It could be repressive elements that cause a reduction in activity. What this results in is limited reproducibility, and that's what you see precisely here. This is this example shows 3 embryos injected with the same forebrain enhancer. You see that all of them have forebrain activity, but you see that the amount of activity in the forebrain varies across these 3, and that there is various ectopic staining in each embryo. This makes it harder to interpret this. You need to look at a larger number of embryos to be sure that the pattern is reproducible. And it makes it especially hard to look for more subtle changes associated with allelic variants of the same enhancer, where you're looking for changes in the in vivo activity. So a few years back, we decided to improve on this assay. And here is an improved version that we have been using for a few years now, which is an assay that builds on this but uses site-directed integration to generate these transgenics. So it's effectively a knock-in assay for Lexi reporter activity. The way this works is we take the same construct, but we flank it with homology arms that target a safe harbor locus. We tested different loci. The H11 locus was the one that worked best in our hands because it essentially never generates ectopic staining. So it's free of both repressive and activating elements, and it gives you really clean results. So this is the same enhancer shown here, injected with this new type of approach, and you can see here highly reproducible activity, absence of ectopic staining, and because it's CRISPR enabled, you get a much higher transgenic rate, which means you need to inject way fewer embryos, which overall boosts the efficiency of this. It allows you to look at larger numbers, and it gives you more accurate results, bringing the more systematic testing of alleles within reach. If you're interested in the technical details of this, or want to adopt it in your own lab, we put out a methods paper shown here that has all the individual steps and reagents that you need to use for this. I don't want to talk about the method in too much more detail, but instead show you how we can apply this. And I want to show you some data from the initial demonstration study that was led by Evgeny Kwan, where he tried to do precisely that, demonstrate how with this improved assay, where we can look at variants at scale, we can now get a better sense of the functional impact of human variants. In this case, he looked at one of the enhancers that is, well, I'd argue it's probably the single most studied enhancer in the human genome, which is the ZRS enhancer. This is an enhancer that is highly conserved in vertebrates. You find it conserved all the way down to fish. It's really well conserved between human and mouse. It's special in a number of ways. One remarkable thing is that it is located at a really large distance from its target gene, which is sonic hedgehog, both in human and in mouse. It's about a megabase away from the target gene. But it's also remarkable because there is a large number of documented point mutations in this enhancer that actually cause a phenotype that you can very easily measure in experimental settings, which is a limb phenotype. So normally this enhancer is active just in the anterior part of the developing fore and hind limb. That's shown here schematically. So it drives sonic hedgehog expression here in the back part of the limb. But there are many point mutations in this enhancer that cause this ectopic staining at the front end of the limb here, this anterior domain that you see here. This is showing just a Lexi reporter, but when this happens in the context of a real genome and sonic hedgehog is misexpressed here at the front end of the limb, what happens is that you get a digit outgrowth phenotype. So the limbs will look something like this. So this is a mouse limb towards the end of the gestation, so at E18.5, and you can see here the 5 regular digits, and you see here this extra thumb-like digit. So this is what it's called preaxial polydactyly, an extra digit at the anterior end of the limb. And what is interesting about this is that there are many cases from human genetics where this has been reported, but there are also various other vertebrate species that have reported mutations in this enhancer that cause polydactyly. Probably the most famous example of this are the so-called Hemingway cats. They're named after the writer Ernest Hemingway, who had a whole colony of cats that have this phenotype. And the phenotype is that they have 6 digits, as you can see here in this one example. There are other species, such as rodents and chicken, that have this. And what you see here is that the other remarkable thing about this is that there's a large number of these mutations that come from human patients. So what you see down here is the entire length of the enhancer. It's about 800 base pairs long. And each of these blue and yellow tick marks are mutations that have been observed in polydactyly. You see here with these little animal icons, different animal species that have known mutations that are associated with polydactyly. But then all the blue tick marks here are various positions where patients with polydactyly have been reported to have typically point mutations in this enhancer. And if you look at the literature, all of these have been implicated in polydactyly. But then if you look at the literature a bit more closely, what you see is that there are some cases where this is really well demonstrated to be causative, for example, because you're looking at an entire pedigree where the mutation segregates with a phenotype. But there's a larger number of cases where the data comes from a single mutation or like a small pedigree, where it's really inferred from the fact that this enhancer is known to cause this phenotype in principle, and there's a mutation observed in the patient. So there may be some ascertainment bias here, right? So the causality of many of these mutations remain unclear. So we tried to figure out if we can now use our scalable assay to distinguish causative from non-causative mutations. So what we decided to do is to build a large series of allelic versions of this enhancer. So we took the human allele, which we know works fine in the mouse, and then against the context of the human allele, we introduced one by one every single mutation that is known in humans or in animals, and then tested each of these in our transgenic mice, looked at multiple embryos, and assessed whether or not this ectopic anterior domain is present or not. As you can imagine, this is a very large number of mice that we had to look at. And here in this one single slide, a large number of experiments is summarized. And this view already shows you by the outcome, like organized by the outcome, what happened. So in about close to 60% of the cases, we observed that there was ectopic activity when we introduced the single point mutation. You see here, sometimes stronger, sometimes weaker, but in all cases, there's some expression in the anterior domain, suggesting that all of these human elements or human alleles are truly causing this ectopic expression. But then we also found quite a few of these published elements, as well as some new variants that we had received from clinical collaborators that actually didn't cause this, suggesting that these are false positive in our findings. So, and this was quite interesting because it suggests that we can actually distinguish these. You will also notice that most, but not all, of the animal variants cause the polydactyly. These down here I would take with a grain of salt, because remember that we introduced these variants against the background of the human mutation. So we did not test the actual animal version. So take these with a grain of salt. But the important point is that for the human ones, we have now a pretty good prediction of what is causal and what isn't. Now, you might say, well, maybe your assay just is not sensitive enough to pick up residual activity. So we also wanted to do a truly functional validation for this. So if you first, as a positive control, build for a bunch of these enhancer alleles that we had predicted to be pathogenic because they have this ectopic expression, we built proper knock-in mice. So we built a mouse where we replaced the central part of the mouse enhancer with the human enhancer, and then introduced these human mutations that we had validated as pathogenic in the assay. when we did that, in all cases, we observed a phenotype that looked like this. So various degrees of polydactyly, meaning that these are truly sufficient, these point mutations are sufficient to cause the phenotype. So that's nice, but it is pretty much expected. Now, the interesting part is when we take those that we predict to have no impact, what happens when we build those? And indeed, what we observed is that in none of the cases that we looked at, there was any effect on the limb morphology. We didn't see any polydactyly. So what this means is that the assay appears to predict quite well what the actual in vivo outcome is. Another aspect of this that we looked at is, what about all these other positions within the enhancer? So there were about 40 or so positions that had been reported as causing polydactyly in in humans or mice or any other animals. But then most of the positions have never been observed to be mutated in a human patient. But then knowing that there are so many positions that you can mutate and they cause polydactyly, we asked, are there more pathogenic mutations that have yet to be identified in patients? Now, this left us with about 750 additional positions that had to be tested. We didn't quite want to build 750 mice, so we decided to use a combinatorial design scheme where we combined multiple mutations in the same construct. So we took the entire length of the enhancer, which is 800. We excluded the 40 or 50 that we had tested individually, and then built alleles where we combined multiple mutations. We did two different series of alleles, one with a 2% mutation rate, so you need about 43 alleles to do that, and then another one with a more severe mutation rate, 17 alleles that had 5% of the remaining residues mutated. In combination across this series and across this series, every single position was mutated at least once. So when we look at the result, one thing that we observed is that the 5% mutagenesis was a bad idea because in most cases, this was just too much. It actually killed the enhancer. In all cases, we saw either complete loss or reduced activity relative to the wild type, suggesting that we just broke the enhancer and mutated some basic nucleotides that are required for it to be functional at all. And of course, you probably won't observe a gain in function if the enhancer doesn't work at all. But then in the 2% mutagenesis series, we did observe, again, some cases where we broke the enhancer with reduced or lost activity, but we also observed some that were normal, and then we observed several that had this kind of gain of function. Now, this is probably an underestimate because there may be additional ones that have been covered up by losses of function due to other mutations. But what this means is that there are multiple additional positions within this enhancer that have yet to be found in a human patient. And this is, I think, quite intriguing because it means we may have now data where we can wait for human genetics to find any such mutations and see if it confirms our predictions. So in conclusion, I've shown you one application of how we can use this larger scale assay for assessment of variance at scale. I've shown you how when we did this for the 0S mutations, that we did find many that are truly pathogenic, where we now have experimental validation that these are pathogenic enhancers, but we also found many that were not pathogenic when we tried to validate them. I think this is an important reminder that we need to be very cautious when interpreting rare variants, and also something to keep in mind as we get better at developing in silico methods for predicting the impact of mutations. I believe we always need to validate them experimentally to be sure that we have correctly predicted a potentially pathogenic variant. And then finally, I've shown you from this example how we can potentially predict additional pathogenic variants before we observe them in patients. I will say that this comes with a bunch of caveats. importantly, this is a very easy to assess phenotype, which is why we did it. For many other organ systems, developmental processes, this will be much harder to do. But I think it's an intriguing perspective to think that we might be able to get experimental validation before we even observe a mutation in a clinical setting. With this, I want to switch gears and talk about another study that also uses this assay, but it uses it for a different purpose, which is, again, looking at the idea of like, now how can we get better at understanding how enhancers work, which is, so looking at the functional anatomy of in vivo enhancer. This is, or this was a study that was driven by Michael Kosicki, who just wrapped up his postdoc with us, and this was published just a few months ago. So what this aims at is trying to understand better how enhancers really work. So what you see here is the conventional models of how we imagine enhancers work. So they're a stretch of DNA that contains binding sites, and then a bunch of transcription factors bind to this DNA. And what we know is that these transcription factors are there, but what we generally don't understand for any given enhancers is which of these binding sites are truly important for function. We do know that any given enhancer typically has binding sites for multiple different transcription factors, so there may be combinatorial effects. We also know that many human enhancers have redundant binding sites for the same transcription factor. We typically don't know what the significance of that is. Are they functionally redundant? Do they add up? Typically don't know. And generally speaking, we don't know how the different parts of the enhancers, the binding sites, possible other components that we don't appreciate yet, how they relate to each other to give rise to the overall function of the enhancer. So we decided to look at this more systematically using a tiling mutagenesis strategy. So the idea was that we use an actual in vivo assay to look at the functional impact of mutations. So we took a number of enhancers and then first made them smaller to identify the core of activity. We then built these tiling series where we went through each enhancer and then in 12 base pair blocks. So this roughly corresponding to the size of a larger transcription factor binding site. One at a time, we would mutate all of these blocks, and then for all of these alleles, build transgenic mice. And the idea was that when we do that, we can then assess the outcome. And by doing so, reconstruct the code, meaning we can assess which of the subregions of the enhancer have an important function versus which of them are not required for in vivo function. And then from this, we may also be able to look at combinatorial rules, test combinatorial patterns to see how different enhancer mutations add up. So here's what we did in reality. So we picked 7 enhancers. 1 face and limb enhancer, 3 different heart enhancers, 3 different brain enhancers that you see here. After making them smaller, they were between 220 and 430 base pairs in size. If you add this up, it gives you a total of about 170 12 base pair blocks that we had to mutagenize and then test. And so then we generated for each of these alleles multiple transgenic embryos and assess the activity pattern. So the way we do this is we generate the embryos and then ask annotators who are familiar with these kinds of assays but are blinded to the genotype of the allele that went in to annotate these outcomes against the original wild type version of the enhancer. So possible outcomes could be that it doesn't change the activity at all. So this is all in reference to this particular enhancer. So it might not change the activity at all. It might cause a partial loss of function, a complete loss of function, or you might see a gain of function, meaning activity in other parts of the embryo where the enhancer is normally not active. So this was, again, a large number of transgenic experiments. Let me jump right to the ultimate outcome. So after testing all of these, what we found was that most enhancers break relatively easily. So if we add up all the 167 blocks that we looked at across all the 7 enhancers, we saw that of the 12 base pair tiles that we tested, only about a quarter or so caused no change at all. Another quarter caused a minor loss of function, but then about half of these caused a major loss of function, a complete loss of function, or a gain of function. This is what it looks like when you look at the individual enhancers. So here's each of the enhancers that we looked at. And along the length of the enhancer, you see the 12 base pair blocks. And then in the same color scheme, you see what happened when we mutated each of these. So you can see two things. So one is you see that these functions are generally distributed across the entire length of the enhancer. The other thing that you can see that there are some enhancers that appear to be very fragile for some reason. So for example, these first two, where almost any block that you mutate causes a change in a function. We have others that are more robust, like these down here, that have many of these dark blue tiles that you can mutate and just nothing happens. But overall, the majority of these enhancers had at least one of these, what I call Achilles heel tiles, meaning a single tile. If you just mess with this one tile, you will completely kill the enhancer. And all of them had multiple of these light blue blocks, which are major or full loss of function. So all of these enhancers have multiple ways in which you can break them. From this, we also started to explore in some more depth the logic behind this, right? How different subregions of the enhancer interact. So for example, we took, again, using this enhancer as an example, two different mutations that individually cause a minor loss of function or a major loss of function. We combined these two block mutations in the same construct. When we did that, we got a result that just looks like the weaker of these two patterns. So there seems to be no real interaction between these two in terms of function. We observed some where we observed a compound outcome that was more severe than the individual ones, like this one. So this is the more severe one across these two. When we combine them, so this one has these two dots. I hope you can see this over Zoom. This dot disappears, so it indicates that this one is more severe. And then we had some where, for example, these two that were individually a major loss of function, where when we combine them, you see a complete loss, indicating that these may be functionally redundant. Now, when we did this systematic screen, we intentionally did this, selected these 12 base pair blocks agnostic to any transcription factor binding sites that we knew or suspected were in there, to get a clean and unbiased view of how the function distributes across the length. But of course, in the end, we know it's transcription factor binding site. So we also looked at this in some more detail and tested, can we use this to narrow down what is actually happening at the transcription factor binding level. And so an example of that is shown here. So you see here at the top an enhancer that in its wild-type version is active in the developing heart. So you see here a front view of the developing heart. It's active in the ventricles and the outflow tract of the heart, but not in the atria. For this particular enhancer, when we did the block mutagenesis, we observed two blocks, this one here and this one over here, that when we mutated them, caused these changes where the activity, specifically here in the left ventricle, was completely abolished. In other parts, it was retained. And when we look at these sequences in more detail, we see that these two actually correspond to well-known heart development transcription factors, GATA4 and MEF2C. We can now hypothesize that it's these particular sequence motifs that are disrupted. So we can just take the largest letter in each of these, presuming that this will kill the transcription factor binding site, and just introduce a point mutation into the wild-type enhancer. And when we do this, we see essentially identical outcomes, indicating that these are indeed the transcription factor binding sites. Something that I don't have time to talk about is that we also went into some depth in assessing the use of the latest generation of deep learning models for making sense of those parts of the enhancer that are not obvious transcription factor binding sites. In particular, we collaborated with Anshul Kundaji to have a closer look at how Crom BPNet does this, and it does overall quite well. And I don't have time to talk about that aspect of the study, but if you're interested in that, the paper is out now. So I encourage you to have a look at that. So in conclusion, for this study, we tested about 260 constructs. In total, we had to look at more than 1,500 mouse embryos. To me, the single most interesting finding was that you can break these enhancers in so many ways, which is different, I think, from at least my preconceptions that enhancers have just like a small number of transcription factor binding sites that you mess with, and messing with any of them will break it. I think there's a large put it the other way around. So there's a large number of sites that you can mess with, and they will disrupt the function of the enhancer. Another unsolved problem is that we saw quite a bit of variation in the robustness of these enhancers to mutation. We don't quite know why that is, but that would be something that is really important to understand. And I've shown you how we can use this data as a starting point to look at the actual sequence motifs that are embedded in these enhancers. It provides a way to get to specific transcription factor binding sites. We can also use it to benchmark deep learning models to assess their value for predicting how a given mutation changes the activity of the enhancer. Now, Stefan, I see that we have 5 minutes left. Should I still do the last part, or should we go to Q&A?
If you can summarize it, maybe. I'd love to give a bit of room for discussion still.
Okay, I can very quickly summarize it. The last one
Thank you.
is about gene deserts. The human genome contains these vast gene deserts that have been observed for a long time. They don't occur by chance, they are conserved, but it's unclear what exactly they do. There are more of them than you would expect by chance, indicating that there's something to what I like to call desertness of these regions that prevents the insertion of new genes, but it's not quite clear what drives it. These were first observed when the human genome was sequenced, but what remained unclear for a long time is to what extent they harbor important functions, because there were early studies indicating that they contained a lot of enhancers, that they contained a lot of conserved non-coding sequences, but there were also early studies that knocked out just 2 of these deserts and just nothing happened. So was unclear. So what we decided to do is revisit this, and again, now that it's easier to knock out large parts of the genome using CRISPR, to knock out a larger panel of 10 different gene deserts in the mouse genome, to just get a general sense of how common removing entire gene deserts, how commonly that causes any significant phenotypes. Going through the results very briefly, what we found was that of the 10 that we tested, 4 had viability phenotypes. 3 of them caused pre, peri, or postnatal lethality. Another one caused lethality around weaning due to reasons that we don't quite understand. Here's just one example of a phenotype caused by this. In this particular example, it causes a phenotype that resembles the phenotype of knocking out a nearby gene. So in this case, it's pretty clear what happens. There's probably some regulatory activity in the desert that is required for normal expression of the gene in development. But in most cases, the answer is less obvious. And even in this case, we haven't been able to find the specific answer. It's hard because the desert is so large. For the other ones, we are still in the process of phenotyping them. We have expression phenotypes for another 3. We have anecdotal phenotypes that we're still revisiting at larger numbers. The bottom line is that in the majority of cases that we looked at, we saw clear viability or at least expression phenotypes. And so jumping straight to the conclusions, these deserts, so I would argue that these deserts harbor critical functions. Many people like to stay away from them because they're hard to deal with simply because of their size. They're unwieldy. They contain all sorts of stuff, I would say, in terms of non-coding functions. But collectively, these deserts are not like weird one-off phenomena. Collectively, they make up about 25% of the human genome. And I would argue that we need to look at them carefully as a major reservoir of potential pathogenicity, knowing that many of them harbor these critical functions. Something that would be really important to understand is which of the deserts contain critical functions, and which of them don't. I think this would be a very important piece of information for interpreting, for example, medium-size indels that we see in human data. If we see larger or not, like indels that fall entirely within a desert that we know is not required for viability, they are much less likely to be causative of a phenotype that we see in a patient. At this point, we don't know yet how to exactly distinguish those that are causing a major phenotype versus those that don't. One potentially attractive idea could be similar to the gene knockout project, to just use the mouse as a model to go through the few dozen or a few hundred largest deserts in the mouse genome that are conserved to human, to get a first map of this and to guide interpretation of human data. And then there are all sorts of other evolutionary questions around like why do they exist in the first place? But I'll leave that for another day. And with that, I will also, in the interest of time, not read out the acknowledgements. I just point out that a larger number both within our team have been contributing to these studies, as well as a bunch of collaborators down here.